All right, hey everybody. We're gonna do this last video and have fun with it, I guess. Okay, so group problems for number 1. We're doing Sager sequencing, and the best way to do that is to just learn how to do it, I guess. We'll teach you how to do it. So it says, it gives you this thing, 5' over, and then it says primer annealing site. So it says, what would the banding look like on the gel? So you always start with the primer annealing site, and that's going to be the smallest fragment. So the G and then nothing after that because of a DDNP, like a DDGTP anneals to that, or a C, I guess, anneals to that one. That is going to be the lowest one. So it kind of looks like this. I don't know if you can see this. It's going to look like that. So if you start from the top, from the primer annealing sequence, you just do the complementary strand, and that's what would anneal to it. So for the G, the first G, a C would anneal to it, and then if it's a DDCTP, it's only going to do that, and nothing else can bind to that and keep going along it. So that would be the lowest one right here, and then you just keep going up along it for each sequence. So that's how you do that first part. And then for part B, it says suppose you use E. coli DNA polymerase 1 during the sequencing reaction instead of Taq polymerase. That would not let the reaction work because, let's just make sure, would not synthesize DNA because Taq polymerase is found in like the bacteria that are in the thermal hot springs, so they can survive super hot temperatures. But E. coli DNA polymerase have to, they'll be denatured to basically kill it, so it won't work. So nothing can polymerize or put on the new nucleotides. So for C, the helicase would not help or harm. It wouldn't do anything to the reaction because helicase unwinds the DNA, and so does the heat of the first step of PCR. So it would kind of just be there. It'd probably denature helicase, but it wouldn't do anything to the reaction. And it says suppose that DDATP, you added that to one of the 4 tubes, but forgot to add the other 3. Assume that the other components used for sequencing are normal. So everything would run correctly, but since you didn't add the DDGTP or the T or the C, it would keep going if you had a bunch of T's. Let's say your primer annealer started right here and they had like T, a bunch of them. That would be able to put a bunch of the A's on, but the second, it would only end at those ones basically. So like A would go and it would end and end and end. But once you had different ones, it would just keep going because there's no deoxy nucleotide that could stop it. So you wouldn't really be able to sequence it. That's how you do number 1.
Okay, so number 2 is the fill in the blanks. For A, it says during a typical PCR cycle, the DNA synthesis step is done at 72 degrees Celsius, while the annealing step is done at 55 degrees Celsius. B, it's the AMPR is a common selectable marker that is found in many plasmid vectors, while restriction recognized in cleaved DNA molecules at palindromic sites. C says during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive electrode during this electrophoresis. DDATP, it lacks the 2' OH group and the 3' OH group. E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. During blue and white screening, the ITPG is commonly used to induce the expression of the black operon, while the XGAL is cleaved by beta-galactosidase to turn E. coli cells blue. And for number 3, it says that we are interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. The sequence to be copied is located between the sequences in the bold below, so it gives us 2 different strand. And A says write the single-strand DNA sequence that you would use for the 2 primers used in the PCR experiment. Note that each primer should anneal to one of the sequences in bold. So for the reverse strand, we're going to start 5' and it goes CAG, TC, TGC, AATT, GAGG, CT, 3', and that's where it cuts off. And the other one is for forward strand is going to be 5', GAG, CCTT, CACT, CAG, TC. And then for B, it gives the equation that helps us to determine the annealing temperature that should be used for PCR. And so that is 81.5 plus 0.41 times the percent minus the 675 over N. And it gives us N as the number of nucleotides within the primer. And so it gives us these, based on these two strands, it asks, or we're supposed to find the temperatures that we predict for the primer for them. And for the forward strand, we find that percent GT or GC is 56%. And so then we plug that into the equation up at the top along with the N, and that gives us 62.3 degrees Celsius for the temperature. And for the reverse strand, we find it's 50% GC. And as we plug that in with the nucleotides as well, we get 59.8 degrees Celsius. And for C, it says each PCR cycle contains a DNA synthesis step. What are the sequences of the two newly synthesized strands of DDCTP is accidentally included in the PCR experiment? Assume that DATP and DTTP and DGTP and all of the appropriate PCR components are found in the reaction mixture. And so based on these from our reverse strand, we're going to get 5', GGGATTAGC before it cuts off. And for the forward strand, we're going to get, starting at 5', CTAATTT before it cuts off. And D, when we learned about DNA replication in E. coli, we learned about DNA A and DAM. Why are these two proteins not included in the PCR experiment? And so for these reasons, the Taq replication doesn't need these proteins to replicate. And the other would be, like Nathan was saying, with the thermal springs or whatnot. The DNA would, or these proteins would, what's that word? Denature. They would denature in the heat that is needed for that.
Okay. So for number 4, this one's pretty, I don't like these ones, but the first one's just make a gel. So you run an agarose gel. I'm pretty sure it's agarose. Yeah, gel agarose. Electrophoresis. And, oh, it does say make a ladder. Well, I forgot to put a ladder on mine. I did, but I didn't do it with the same markers. So my bad. But basically, you look at each one of these and you say reaction 1 was cut with BAM, reaction 2 was cut with EcoR1, reaction 3 was cut with Hindi 3, and and then 4 was cut with 2 of them, 5 was cut with different 2 of them, and 6 was cut with different 2. So you take that. So if 1 was 50 base pairs, you would make a gel that looks something like this. And then you say, oh, 50 base pairs, 350 and 550 are for reaction, or 500 are for reaction 1. So 50 is way down here on mine for reaction 1, in between 25 and 100, and then 300, and then 500 is right up here. And then for the next one, you just keep doing that. You kind of figure out how far down it would migrate. The most important thing to remember is the least amount of base pairs or the smaller size of DNA is going to migrate the furthest down an agarose gel. So you just keep doing that and kind of use the ladder that she told us to use, the 100, 200, 300, 400, 500, 1,000 fragments, to kind of estimate where those would be. For B, it says draw a linear restriction map. So she did a video on how to do this, and the best way she showed was to take each reaction 1 through 6, and draw out what the linear would look like. So let's say if you have, it's this long, and then for the first one it was cut twice, so you're gonna have 2 spots where it's cut, and you do one with 50 base pairs, one with another amount. I think it'd be like 300, and then the other one with 500, probably 250 actually. And then you indicate how big those are. So you do it for each one, and then you can see kind of along the line which one. So for that, we're gonna have a 50 base pair first, and then the BAM H1 is gonna be cut, and then 150 base pairs, and then the EcoR1, and then 100 base pairs, and then the Hindi 3, and then another 100 base pairs, and then the BAM H1 is gonna be cut, and then 500. So then for C, it says predict the fragment lengths if the linear DNA molecule is cleaved with all three. So like in reactions 4, 5, and 6, it was cut with 2. So now it's kind of like, what are your fragments going to be if you cut it with all 3? So look at your map, you'd see that there'd be a 50 fragment portion, a 150, 2 100 fragment portions, and then a 50 or a 500 base pair portion. So if you're in them on a gel, a 50, a 100, a 150, and a 500 would show up, but there would still technically be 5 fragments.
So for number 5, so we're studying a gene, our favorite gene, that you've identified in the yeast Saccharomyces cerevisiae. Says detail the steps that you would use to generate cDNA from this mature mRNA, clone a CDA version of your favorite gene into a plasmid vector, and use blue-white screening to identify plasmids that contain the YFG cDNA. So the steps, the first thing that we're going to need to do, we're going to need to generate the cDNA. So basically we need to isolate the mRNA for the favorite gene. We need to add a poly-DT primer, reverse the transcriptase, use RNase H, and then add DNA polymerase and the ligase, and that's going to give us the cDNA. And then cloning that into a vector, or into a plasmid vector, is the next step that we're going to need to do. And so with that, we're going to need a restriction enzyme. The cDNA and the plasmid are going to be mixed, and the hydrogen bonds of the sticky ends are going to,
Anneal
back together. And then the DNA ligase is added to create a recombinant DNA molecule in which the cDNA is inserted into the cloning site of the plasmid. And the last thing, we run the blue-white screening to identify those different plasmids. And so the E. coli cells are going to be transformed with the recombinant DNA molecule. And then those are going to be plated on an agar plate that contains the ampicillin, the IP, TG, and the X-gal. And then we identify the white colonies, then we identify the blue colonies, and then we use the restriction mapping to identify the recombinant DNA molecules. And that is it for videos.